This is officially still Chapter 1, Introduction to Microbiology, although it is the second week and the second set of lectures. So we kind of move a bit slowly in the beginning, which is probably a good idea as everybody gets organized and gets on to mastering microbiology and the textbook and all that's involved in gaining access to this course. So please contact me if you're having any issues, and I'll do my best to help you out. So today, what I want to focus on in the remainder of Chapter 1 is talking about culturing microbes. We did a little bit of historical perspective last week and just sort of an overview of how we think about bacteria and what they're all about. But in order to study a microorganism, it's absolutely essential to be able to culture them. In other words, you have to be able to grow them in a controlled setting. And you have to be able to grow the microbe in a pure culture, where we define a pure culture as consisting of only one type of microorganism. And this way we can characterize the microbe, we can determine its properties, and see whether or not it is important from a medical perspective, for example. So going back to Robert Koch, who was one of the pioneering microbiologists, he looked at the idea of assembling a research team in his laboratory to use agar, which was a sugar, I guess a sugar polymer, and it forms a jello-like texture, and on the surface of which one can grow bacteria. So you're going to be using agar all the time in the laboratory with various nutrient media to culture different types of microbes. So it is ubiquitously used to study bacteria and to obtain pure cultures. Also, you'll be using the Petri dish invented by Julius Richard Petri in Koch's lab in order to conveniently look at the bacteria and prepare them and incubate them for growth properties and to analyze them. You'll also be learning in lab about different type of media which can be suited for one type of microbe or another. So here we see the first diagram here looking at the different types of ways in which we can culture bacteria. We can use solid culture, such as this agar plate, a slant, which is sort of a modified plate. It gives you a larger surface area than simply a straight area across. So this gives you a greater area to prepare cultures. And then over here we have a broth, which is liquid culture. So liquid culture is okay to use when we have pure microbes, pure cultures. But if we want to obtain a pure culture, then we want to separate out the different strains by forming individual colonies, whereas a colony of bacteria consists of the outgrowth of a single microbe when they're plated properly. And then also, depending on the other requirements of the bacteria, they may require oxygen or they may not. So you might use an agar deep if you want to keep the microbe away from oxygen, which can be toxic to certain bacterial strains. Another very important aspect, which we will be going into in greater depth, both in lecture and in lab, is the idea of using an aseptic culture technique. In other words, in order to obtain a pure culture, you really have to get rid of or exclude all other forms of microbes besides the one you're actually interested in studying. And that requires using aseptic technique. So you want to make sure everything is sterile that you're using, the culture is sterile, the dishes are sterile, the test tubes are sterile, and the various objects that you use for preparing colonies. So all of this needs to be sterile, and it can be very time-consuming to prepare all of these culture media and the various tools and instruments and cultured dishes that one needs to carry out all of these experiments, but they're absolutely essential. So we want to emphasize safety in the lab, and your lab instructors will be doing that. Both in order to maintain a aseptic technique and also in order to maintain your health and safety. Although you will be using non-toxic cultures, it's still good practice to use sterile technique for your sake as well as for the sake of the work that you're doing. Now, this is called a streak plate, and you'll be studying this in the lab as well, where you take a more concentrated, um, I guess, glob of bacteria, and you spread it out on a dish, and then you flame the needle, and then you put it back in and continue just taking a tiny bit from the first section, spreading it out again, then flaming it and taking another tiny section, and you continue to do that to generate essentially a serial dilution. So hopefully by the time everything grows up, you've got single colonies, which can be distinguished from all other colonies, and that represents what we would call a pure culture, a group of microorganisms of the same strain that originated from a single cell, and that's called a clone. Now, in the bottom panel, we are looking at a mixed culture. So sometimes we don't have a pure culture to start with. So we're going to streak out a plate, and we'll obtain perhaps several different types of bacterial colonies. And based on our knowledge and understanding of the properties of the colony, we can then select just one colony and then grow that culture up as a pure culture to separate it out from all the other cultures and colonies and types of bacteria that may have been in the original mix. So you'll be doing this in the lab. Now, in addition to various streak plate and culture techniques to cultivate bacteria and to obtain a pure culture, we also want to analyze that culture for the type of bacteria that's present. And there are lots of ways of classifying bacteria. And one of the most simple and important really is the use of various stains and dyes, which can give rise to unique colors or patterns of cell formation, or combinations of both and other characteristics, in order to distinguish one bacterial type from another. And this is differential stains are a very, very important way of studying microbes and classifying them, and also to verify that you have a pure culture after you've prepared one, and then certainly in diagnosis as well, when we want to determine the causative agent of a specific infection. So some of the stains, as you can see here, the most common ones are called basic dyes, which means they're positively charged, and they will therefore interact with the cell, which is negatively charged. So it's sort of an electrostatic interaction. Some of these include methylene blue, which gives a deep blue color, crystal violet, which gives a purplish blue color, safranin, which is red, and malachite green, which obviously is green. Sometimes there are acidic dyes that are used. So you'll be seeing some of that later on in your staining procedures. Now, in addition to using a stain or dye, we may also want to use what's called a mordant. So a mordant is one that helps to facilitate the staining procedure. So it helps to allow for a stain to be more effectively or efficiently taken up by an organism, and can be a very important part of differential staining techniques, and we'll see how that works later. Okay, so simple staining is what we would call simple. It's just using simply one dye, okay? So using a simple stain, we might want to just stain the bacterial cells individually and look at them under the microscope so we can see what their approximate size is. And it's easier to do that when we've stained the surface of the cell rather than just looking at them in the native configuration. In addition to the overall size, we can look at the shape. And as I mentioned to you in last week's lecture, the shape of the bacterial cell is very, very important in its classification. And we talked about the fact that there are three basic shapes. There's the spherical shape, there's rod shape, and then there's a spiral shape. And there are variations on each of those themes, but using a simple stain can help us to facilitate the actual shape and size of the bacterial cell. Also, very important to look at cellular arrangements, because both in rod-shaped bacteria and spherical bacteria, there can be various arrangements. In other words, the cells may remain stuck together after cell division, and this can help us to classify, because certain types of bacteria have a tendency to form certain cell-to-cell arrangements. So we'll be talking about that. Now in lab, you're going to be using simple staining techniques, so I'm not going to spend a lot of time talking about it, but just quickly, the first thing you really have to do is you have to heat fix a smear. So you take a needle and you take the O-loop basically, and then take some bacteria from your culture, either liquid culture or a dish, and then you would spread that as a smear over the slide. And then you would heat that, okay? So you run it through the Bunsen burner a couple times. It's called heat fixation. And that causes, the heat causes the cells to stick to the surface of the glass so they don't get washed away during the staining procedure. Then you add some sort of dye, in this case, for example, methylene blue. Give it enough time so that the cells can take up the dye. Then you wash the excess dye off, and then you put a cover slip on and take a look. And that's when the fun begins. You get to look under the microscope at all the wonderful and amazing different arrangements and sizes and shapes that bacteria can form. So this is, in figure 1.12, this is a methylene blue stain of E. coli. You see that they're fairly small rod shape, and they are stained here a deep blue based on the stain that was used. And it's much easier to visualize these as they are stained rather than just trying to look at them individually. They kind of are more transparent in the unstained configuration. Now, there are certain stains that we use to identify structures that are found in the bacterial cell. And this part of this gets into the whole structure of the bacterial cell and what kinds of shapes and structures and appendages they have. Now, an important characteristic which can be used for classification in bacteria is the presence of flagella. So flagella are whip-like structures that can be found in both eukaryotic and prokaryotic cells, but they are structured somewhat differently in the two systems. And the flagella are used for motility, and they can be very helpful in classification. So if you're dealing with a rod-shaped bacteria that is classified in the book, so to speak, as having flagella, and you're trying to make a differential diagnosis, you'd be looking for a rod, and you might stain with the flagellar stain to make sure that it actually has flagella to confirm your prediction. In addition, the spirilla all have flagella, so it becomes a very important tool for identification and study of the various structures of the bacterial cell. So here's a couple of specific structural stains. Here's a flagella stain, and here you can see the multiple flagella coming off of the rod-shaped bacteria here. Now this is a stain for a capsule. A capsule is essentially a polysaccharide envelope that surrounds the bacteria. For certain strains of bacteria, it can be very, very important for infectivity. Talk a lot about capsules later on. And then here is a stain for an endospore. And endospores are found in certain types of bacteria, and they're very important because not only do they play a role in how the bacteria maintains itself and how it is cultured and proliferates, but also it's very useful for classification because certain bacteria have endospores, and if you're making, again, a differential identification, you should be able to document that the particular strain that should have endospores actually tests positive for them. So you can see that you can do quite a bit of structural assessment by using the various types of structural stains. And then this just goes into examples of this. So you have the capsules, which are, again, the carbohydrate-based structures, and very, very important in adherence and as part of the infectious cycle, which we'll be talking about later. And again, we can use specific stains to visualize more clearly the capsule. You have endospores, which can be stained, okay, as well. And here the example is malachite green, and you can see that in the diagram as well. You can see sort of the greenish staining spores. Here we're using two stains so that we can use one stain that helps us visualize the endospores, and then we're using safranin, a pinkish red stain, to look at the actual morphology of the bacterial cell. Now, two very important stains that you'll be studying extensively in lab are the gram stain and the acid-fast stain. And this is an example of differential staining that's used to divide bacteria into very important categories, very important groups in terms of their structure and many of their properties, including their roles in human infection. So one of these is called the gram stain procedure. And essentially what the gram stain does is it helps us to distinguish two fundamental basic structural organizations of the bacterial cell wall. When we get more into the structure of bacteria, you'll see that the cell wall is probably the most important structural characteristic of the bacterial cell, and it comes in two varieties. They're named after the staining procedure, gram-positive and gram-negative. And the gram-positive bacterial cells will stain positive with this whole protocol, and then the gram-negative will lose their stain as part of the staining procedure, and they're called gram-negative. So those that retain the gram stain are gram-positive, those that lose the gram stain are gram-negative, and it's a very, very important tool for classification as well as even in the study of infectious disease. So you're going to be studying this extensively, and it's really a lot of fun doing the gram stain procedure. So what you're actually doing is taking advantage of that, again, that structural difference. So if you look at the procedure, which we'll go over very fast here, and I can show you the pictures, which is probably more informative at this point. So here we have some bacteria mixed culture, meaning that it contains more than one type of bacteria. And some of them are staining gram-positive, and some of them are staining gram-negative. So what do we mean by that? Well, gram-positive will stain positive with the basic gram stain, which is has a purplish dye. And you can see here, these spheres are staining gram-positive. Now, some of these rods are not purple. They're sort of a pinkish color, and that means they're gram-negative. They don't retain the stain. So the question is, well, what's going on here? Well, let's look at this. We start off with the heat-fixed slide, okay? So we take our cultures, we put a smear on the slide, we run it through the Bunsen burner a couple of times to fix the cells so they don't wash off. And now we're going to start with the first part of the staining procedure, which is we're going to stain the cells with crystal violet. And everybody's going to stain positive with crystal violet. So at this point, we're not doing anything differential. We're just testing everything out. We're adding the stain. Now, the next thing that we're going to do, and this is where we're going to start getting specific, which is we're going to add a mordant. Again, we talked about a mordant, something that helps the cell to retain the stain. So the iodine is a large element, iodine. It is a large negatively charged element, which will bind to the stain and help to trap the stain inside the cell. And therefore, this is important so that the stain doesn't wash out. Now, we're going to take those cells that have been stained with the crystal violet and the mordant, and we're going to use a destaining or decolorizing solution that can have a couple of different ingredients, but most commonly is an acetone alcohol mixture. And what's going to happen is that here's where your differential reaction comes in. Because it turns out that the gram-positive cells, or bacteria with a certain type of structure that's capable of retaining the stain, has a very thick cell wall consisting of multiple layers. So when you wash away the lipid layers with this acetone alcohol decolorizer, you're not going to affect the gram-positive cell wall because it has so many layers of the polymer peptidoglycan, which is unique to the bacterial system. So it's a peptide-based polymer that's resistant to the acetone alcohol, and therefore the cells retain the stain. Now, when you come to the gram-negative cells, these actually have a much thinner cell wall, and therefore, and you wash away the outer lipopolysaccharide or LPS layer, and then the stain will come out too. So you decolorize selectively the types of bacteria or the groups of bacteria that have a thin cell wall that have the LPS layer around it, which is soluble in the acetone alcohol. Now, in order to further distinguish the two, the decolorized cells and both samples can actually be counterstained with safranin, which is a reddish color. And the gram-positive cells that have retained the purplish stain will stay purple because they were already purple. But those gram-negative cells where the crystal violet was washed out by the decolorizer will now stain pink. And here you go. So you end up with the ability to distinguish bacteria that are classified into these two major structural groups based upon whether or not they stain purple with the crystal violet as gram-positive cells, or whether they stain negative and therefore lose the crystal violet at decolorization and pick up the safranin at the secondary stain step. So it's a very, very important and simple procedure that was actually discovered by Gram, hence the name Gram stain, and it's very important clinically. Okay, so very often if you have, here's an example of getting tested, the blood tested for bacteria infection, and what the healthcare professional would want to know right away is, what are we dealing with here? Are we dealing with gram-positive or gram-negative? It's very important for clinical diagnosis, and it's also very important for making therapeutic choices, because some bacteria are going to be much more sensitive to antibiotics that target the bacterial cell wall than others, depending upon the structure of their cell wall and how that maintains the bacteria of the cell, the integrity of the cell. Okay, so you can read this over, which explains very carefully the actual Gram procedure, okay? So they're talking about different types of gram-positive and gram-negative. And here we have, for example, hold on, they're just asking me if I want to keep signed in. Here we have bacteria that would be either gram-positive or gram-negative, staining with the Gram stain, and then you can also use specific stains that are much more specific for individual groups of bacteria. And you're also going to be doing this as part of your lab work. And one of these is called the acid-fast stain, which distinguishes a group of bacteria called the mycobacteria. They're called mycobacteria because they have in their cell wall a substance called mycolic acid, and the acid-fast stain will stain for that. And it's useful, for example, in detecting tuberculosis infections because they're members of the Mycobacterium genus, and also leprosy, Mycobacterium leprae, also will stain positive for acid-fast. But only these specialized groups of bacteria that actually have mycolic acid in the cell wall will stain positive. Okay, and then this goes with through the Zeal-Nielsen procedure. You're going to learn how to do this, but it is a fairly simple and straightforward and very important way of distinguishing the microbacteria from other infectious disease agents, and has great clinical significance. As we talked about before or last week, tuberculosis continues to be one of the most important infectious diseases worldwide. So this shows you what you would look at in the microscope when you've done an acid-fast staining procedure where you're dealing with a mixture of bacteria. Those that are acid-fast are going to retain the stain and test a pinkish color, and those that do not retain the stain will be stained by the blue, the secondary dye. So this is a very important distinction that's made, and the actual primary dye is called carbylfuchsin, and the red dye will be decolorized unless the mycolic acid is present. And methylene blue serves as a counter stain in this case. Okay, So here you have the clinical case, okay? There's a video that you can watch, and I think these are very important. I really like case studies in clinical courses. I think they really help to tie a lot of concepts together, and they're extremely interesting and very practical, so I would certainly recommend that you watch this. Okay, now microscopy. Just a bit about microscopy. Again, you will be using the microscope continuously in the microbiology lab. They don't call it microbiology for nothing. The microbes cannot be seen with the naked eye. They require a light microscope. And most of the bacteria that we actually study can be seen fairly nicely using the standard compound light microscope. And this is a nice photograph showing all the important pieces and parts. You'll be studying that in the lab. This is very important that you know how to use a microscope and how it functions, because it really is your pair of eyes in the lab. It's essential. So I'm not going to go through all the different parts of the microscope. It's explained here very nicely, and you should know what each of these parts does and how they help you to focus on bacteria of different sizes using different types of magnification, okay? And the oil immersion is sometimes useful as well. It gives you a higher resolution, and it is useful for the smaller specimens, smaller size specimens of microbes that you may be studying. Okay, so this goes into comparing light microscopy techniques. All right, so this is amoeba proteus. Amoeba are pretty large, so they can be seen very easily. They're eukaryotic, and it goes into the different kinds of imaging. You can have a darker contrast image using a bright background. Here you have a negative image where the sample itself is light and the background is dark. And so you have variations on the theme, which can give you better resolution depending on what approach you use, can give you better resolution of certain structures due to the differential properties of the light and contrast. Now, you won't be doing electron microscopy in this course. It is a very important tool for studying viruses. So although we study viruses and we learn about their important role in infection, electron microscopy is a fairly complex instrument. The electron microscope is, and the procedure for preparing specimens is fairly complicated. So we'll be looking at electron micrographs, but such as this one over here, but we won't be preparing electron micrographs ourselves. And even with bacteria, although we can see them with the light microscope, we get a lot more resolution using the electron microscope. We can see the structural parameters of the cell wall. Here you can see the flagella and their inner structure much more easily. And so for analytical work, the electron microscope can be very useful, certainly extremely useful for the study of viruses. And here you see two types of electron microscopy. Transmission, which essentially looks at a very thin layer of cells. So you have tools that allow you to prepare a very thin section that allows the light through, and then you stain it, and then the passage of electrons creates a contrasting image that you can visualize. And this is scanning electron microscopy image where you're just looking at the outside. You're not getting to the interior, but you're looking at the morphology or shape and the exterior properties of the microorganism. And the procedure is slightly different, and it does not involve thin sectioning of the microbe. So you can read this over. They both have a very important role in the study of microbes. Again, you should look at the part 3 of the video, and again, it's very important that you understand the applications, and very interesting as well. Now, we can use fluorescent stains, which can be very important in visualization and also in studies of high specificity. So we can use antibodies, for example, that have a fluorescent linkage that will allow us to stain very specific structures within the microbial cell or within viruses. And this is very useful analyzing their structure and function relationships that go into determining how microbes cause diseases and how they interact with their human host. Okay. So again, this section is a very nice little segue talking about the normal microbiome and how that interacts with the immune system and how mistakes in the way the immune system recognizes certain elements of the microbial flora that coexist with humans may actually in some cases result in simultaneous targeting of tissues within the body that share some antigenic properties with these microbes, and it can lead to autoimmune disorders where the immune system attacks systems within the entire body. Okay, so here we have the end, build your foundation. There is an assignment that will be due shortly. The first assignment on chapter 4 is being postponed, so don't worry about it. But there is another assignment that will be due February 4th that you should look at and get started with as soon as you finish listening to this lecture and you complete reading the chapter. So I'm going to conclude now, and I'll be picking up later this week with yet another lecture moving on to chapter 2.